All right, we're going to get started. We have a lot to go over today, so I just wanted to say hi and thank you for joining us for our gene therapy town hall. For those of you I haven't met as yet, my name is Elisa Seeger, and I'm the founder of the ALD Alliance. I lost my son Aiden to ALD in 2012 and started the foundation to primarily focus on newborn screening and also to be a place where we can gather information and share with the community. I'm excited to say Mississippi started on July 1st, so we're now at 37 states that are screening for ALD. So that's an exciting piece of news. And just to talk a little bit about, my experience, I remember the diagnosis so vividly. And of course, being in shock and being a late diagnosis and remembering how we had to make decisions very quickly and we didn't have a lot of information or anyone to reach out to get that information from. And that's one of the reasons as well that I started this organization. We have our annual meeting and then, host these town halls to help with gathering information and sharing it with the community. I know that there's been some questions about the gene therapy, and I have to just stop for a minute and say how grateful I am to Bluebird and their dedication for doing this, because I remember over 10 years ago being diagnosed and, my son being diagnosed and wishing that we had gene therapy as an option. And it took a lot of dedication to get where we are right now. Grateful that the FDA approved it in September of last year, and, so emotional to be able to be a part of Connor and watching him be the first boy to have the FDA approved gene therapy. So we're really grateful that this is an option, and especially for families that do not have a sibling match and do not have a match in the registry at all. So today we'll just talk about, any updates to the gene therapy and the MDS cases that have come up. So just wanted to go over how the format will be today. So we'll start with Rich Colvin, who's the Chief Medical Officer for Bluebird Bio, to talk about the gene therapy. And then we'll turn it over to Dr. Paul Orchard from University of Minnesota and Dr. Christy Duncan from Boston Children's Hospital, so they can talk about their perspective on gene therapy. And then followed by that, we'll go through questions, and the questions that were sent in ahead of time, we have a few of those, so those will take priority. And then we'll open it up to other questions. If there are questions, please send them through the chat, and then we will, ask those questions to the group. So with that, I'm going to turn it over to Rich Colvin, and he will start us off tonight. Thank you.
Hi, first of all, thanks so much, Elisa, for inviting me to speak at the meeting. And so everyone knows, I'm Dr. Richard Colvin. I'm the Chief Medical Officer of Bluebird Bio. Bluebird remains committed to the ALD community and really wants to ensure that patients and physicians can make the most appropriate choice for the patients who are going through this. Gene therapy for ALD is complex, and the decision to treat patients with it is equally complex. We appreciate the longstanding partnership with your community as we work to bring new options and hope to families facing this devastating disease. There's never been an easy time for ALD patients and their families, and today I'm going to update you about the results from the ELICEL program. If we go to the next slide, please. For those of you who are not familiar with Bluebird, I'll provide a little bit of background. We're a gene therapy company that's focused on developing new treatments for severe genetic diseases. Last September, after a decade of clinical development, SKYSONA gene therapy was approved by the FDA to slow the progression of neurological dysfunction in boys between 4 and 17 years of age with early active CALD. Importantly, SKYSONA carries a boxed warning for hematologic malignancy, and I'll talk about more about this risk tonight. At the time of approval, there were three boys who had myelodysplastic syndrome following treatment with Skysona or LA-cell during the clinical studies. Today, the therapy is available at 3 activated qualified treatment centers: Boston Children's Hospital, Children's Hospital of Philadelphia, and Stanford University Hospital. Currently, the activation of University of Minnesota as a QTC is in progress and should happen soon. And as you can also see and probably know, that the treatment of patients with Skysona in the commercial setting has begun. These milestones have only been possible because of the tenacity of the CALD community, and you have really persevered through incredible challenges, and also the courage of the families who participated in our clinical studies. My goal today is to share details on recent safety updates and the steps Bluebird is taking as a result of these events. I'll answer the questions I am able to and explain how and when we will share this information as we continue to learn more. If we go to the next slide, please. Since the approval of Skysona, 2 additional children who were treated with LA-cell in clinical trials have been diagnosed with myelodysplastic syndrome, or MDS. This brings the total number of reported cases to 5 among the 67 patients who were treated with LA-cell in clinical trials. While I cannot provide detailed patient-level information, I can confirm that all of these patients are under care of their treating physicians. I share this news with an extremely heavy heart, and I know that all of our thoughts and best wishes are with these boys and their families as they face this challenging diagnosis. And if we go to the next slide, please. I'd like to talk briefly about the steps Bluebird has taken since learning of these diagnoses. First, it's about informing. And in each case, Bluebird has worked quickly to inform investigators and treating physicians to ensure that any families considering Skysona could be counseled on this latest information. In each case, this update happens within days of Bluebird receiving this initial report. And just so everybody knows, one of the cases was diagnosed in the end of May, and the other one was diagnosed less than 2 weeks ago. Consistent with our established safety protocols, we also alerted the Independent Data Monitoring Committee that oversees the safety of our studies, as well as the FDA. And today we are sharing this update with you directly to ensure that obviously there's a close-knit community, and we want to make sure you have access to consistent and accurate information. And I want to pause here to acknowledge that Bluebird heard from members of the community earlier this year with concerns about data transparency. I want to address this head on and be extremely clear. These cases have emerged literally in the last 2 to 6 weeks, and Bluebird has always communicated with urgency as new safety events arose, and we will continue to do so now that we are approved, though the regulations that govern how we do so are different in the post-FDA approval setting. I'm happy to take questions on this later in the discussion. We're also in the process of investigating the cases. We have worked with urgency to launch investigations into each of these cases, and we are currently in the early stage of this work since they happened so recently, and expect to receive the results of laboratory and clinical tests throughout the summer. In addition to conducting our standard assessments to understand the role of therapy played in this, we are also exploring what factors may increase the risk of MDS and potentially other malignancies in patients who receive Skysona or LA-cell. If we go to the next slide. So what does this mean for patients and their families? Well, despite this new cases, we know that many boys do not have any other treatment options and believe that Skysona remains an important therapy. We are convening a panel of experts to get additional perspective on this risk and ways that we can better understand this risk. This discussion is going to take place before any additional children are scheduled for infusion with Skysona. So if we go to the next slide. And so what comes next, right? So in addition to sharing the steps we are taking related to these cases, I also want to ensure that you understand Bluebird's processes in the post-approval setting and know what to expect from the company moving forward. First, regular monitoring. We're going to continue to monitor all the safety events. And we will also reiterate that patients who have been treated with LA-cell or Skysona in the commercial setting should receive lifelong monitoring for hematologic malignancy. Patients who enroll in the post-marketing registry study are monitored for 15 years, and patients who receive the treatment are encouraged to participate in that study. We'll continue to provide updates And what we will be doing is we'll be providing updates, regular reporting to the FDA, meaning health authorities here, and also to our investigators, as well as the data monitoring committees, and to the IRBs of the sites. Right. So information is communicated differently. Information is communicated differently in a clinical study compared to when a therapy is FDA approved. Monitoring is just as rigorous, but after FDA approval, we also have to consider other things. Like we have to follow new rules and regulations that govern how we share information. And there is some variability in how information gets communicated to families by individual clinical study sites and treating physicians as they follow their own internal guidelines as well. However, we're going to continue to publish our data in whether it's in the scientific clinical literature as well as present at congresses in the future. And thinking about timely information, anytime that a new safety risk is identified or the risk-benefit profile of SKYSONA evolves, we will have an established process to ensure expeditious reporting to the FDA and communicate with investigators and physicians who are treating patients who have received therapy after FDA approval. This is the process we are following now. This does not mean we'll broadly share every update in real time. It means that the community can have confidence, though, that their treating clinicians have access to the latest information, and that new risks or shifts in the risk-benefit profile will be broadly communicated with urgency. While we remain committed to transparency, we'll continue to participate in community forums like this one. Discussions about new data and risks associated with gene therapy in the post-approval setting should happen between families and their care teams. And finally, if we go to the next slide. Perfect. Despite these latest developments, we know that we believe that SKYSONA remains an important option for many families. And we remain committed to the community now more than ever, and we will continue to prioritize patient safety and transparency as we always have. The team at Bluebird is behind you, and we are working to ensure you and your care teams have the best possible information as you navigate these very challenging treatment decisions. Again, I want to thank Elisa for inviting me here today, and now I'll ask Dr. Duncan to speak about how the physicians in the community are thinking about these latest results.
Great, thank you. Miranda, I'm just going to share my slides now, if that's okay. All right. Let's see. Thank you. Let's see, just move this up. So thank you very much for allowing myself and for Dr. Orchard to come today. And I'm presenting slides, just a few of them. And then Paul and I are going to just talk about how we're thinking about these things and hopefully save most of the time for questions. For those of you who I haven't met or don't know, I'm a pediatric stem cell transplanter and gene therapy physician at Boston Children's and Dana-Farber, and have participated in all of the ALD gene therapy trials and the treatments of patients after the drug has been approved. Well, we know that much of the focus tonight is on the myelodysplasia cases, which are, for those who aren't as familiar with MDS, which is a blood cancer. Some people call it a pre-leukemia, some people will call it different phrases, but it's a cancerous diagnosis that requires treatment with an allogeneic stem cell transplant. And so while I think we're going to have a ton of talk about that, we did also want to take the opportunity to update you on some of the efficacy data, so how well gene therapy is working for patients, and knowing that the data we have is still ongoing, the data is still being analyzed, so there are different time points for each of those pieces of data. One of the questions that came in before the meeting asked about what happens after 2 years in the data. And so what this slide is this is showing you in the top line, the LE-cell of the SKYSONA, the commercial name, gene therapy, when you compare patients being alive without major functional disabilities, without myelodysplasia, or without getting a second transplant over time. That's compared in the purple, or sort of the lilac line, to patients who, in the study that was used to compare, received an allogeneic stem cell transplant during that period of time. And so when we look at the 2-year mark, we can see in these patients, and I should say also, these are patients who do not have a matched sibling donor. So I think that's an important distinction to make. We can see some differences at 2 years. And then those differences remain relatively the same. And then you'll see this big drop that I just want to talk about at sort of the 8-year time point. The reason we can't look at the difference in survival, this definition of survival without a major functional disability, second transplant, or MDS, in the allotransplant patients, is that's where the study data ended. So we don't have that data going further out. The reason to see for this big drop right here is twofold. One of it's because when you get to that 8-year time point, when this data was collected, there were only 3 patients who had been out 8 years from treatment. And so of those 3 patients, one of them had MDS and the 2 did not. So that's why that big drop is. I think when we get the new data coming out, or the new data analysis, when there are more patients who've reached 8 years, when there are other patients who've gone longer, I suspect that will change. But this just shows you sort of what that data has looked like from the time of infusion of stem cells up to basically 8.5 years after treatment. The next thing I want to talk about is this group. So the allotransplant no match siblings, and look at this data in a little bit more detail. And so this, again, the same definition. So here we're looking at survival without major functional disability, without myelodysplasia, and without needing a second transplant. And so the 100 is at the top. So if you are alive and you don't meet that definition, this is good. You want to be at the top, and 0 is obviously not that. So what I think is important when people, patients and families are making choices, is to look at a couple of things. So when you compare this matched unrelated donor, we don't even have matched sibling on the side. The matched sibling basically overlaps at all these time points. But if you have a well-matched unrelated donor and looking at this, the efficacy is quite similar across gene therapy overall. And so it's really it's not just not having a sibling donor where you see a big difference. But if you get down here to patients who would have a mismatched unrelated donor, I think you can see that's a pretty significant difference. And so, those of you who've had conversations with stem cell transplanters, Dr. Orchard, Dr. Lund, myself, others, and thought about this, there are more details or things that go into choosing a matched unrelated donor. But if you have that, it is important to know that efficacy data, or the data we have based on this, looks quite similar. Oops, sorry, I got the doctor office, versus that when you compare to mismatch. When you look at the mismatched patients, there really is quite a difference. The other thing, so just looking at this and isolating gene therapy separately, looking at that same definition of survival up to 5 years. So at 5 years from treatment, this is just gene therapy alone. When we looked, there were 17 patients who were available for analysis at this point. We can see that definition's met here. She's at 91%. I think a lot of people have told us they've heard that definition over and over, and there it is. But then when you get to 5 years, about 82% of patients who are enrolled meeting that definition. So that's longer than 2 years, but we're certainly waiting to get more data, and that data is forthcoming beyond that point. And then thinking about the efficacy, because as we all know, it's not just about being alive in many cases, but sort of what does that look like? So these two slides, or these two figures, I just want to orient you a little bit to them. This is we call it a box whisker plot. And so basically the group of people who are being evaluated get divided into quarters. So 25% of the people fall into this long line, 25% into this one, 25% here, and 25% below the line. The line in the middle tells you the middle. So if you take everyone's score, everyone's change in their less score, in the middle is where that line is. So what these figures are showing us in the blue is how patients who got treated on ALD 102, so the very first of the gene therapy studies, compared to patients who got allo around that same time, who would have met eligibility for gene therapy, but didn't get it for whatever reasons, what their change in their less score from baseline. So the numbers on the sides are not the absolute less score number. This is the change, so the change in the number of points. And so if you look at in the blue at 2 years, which is, at this point, this is gene therapy, and this is allotransplant patients. And you look at 3 years, which is here in the gene therapy, and here in the allotransplant patients, there is a statistically significant difference in the change in the MRI less score. So patients who had gene therapy had a bigger change in the less score in this study compared to those who had allotransplant. I think one of the things that's interesting about that data and trying to interpret it is sort of the next steps also, and what does that mean for your function? And so looking at the neurologic function scale scores, they're the same. So there's no difference in the change from your baseline. So if you came in with an NFS of 0 or a neurologic function scale score of 1, there wasn't any statistically significant difference across time points. And this is getting up to this right here, this is 5 years. So even if you look at these early stages, it's similar. So trying to put these two pieces of data together, you can see there's a bigger change in the MRI, but not a bigger change in the patient's individual functioning. And so we have to learn and get more data and try to interpret that, in the context of everything else that we're hearing. This is data that probably people have not seen us present yet. This is updated through July. Many people who are on the trial, I know I've consented many people and consented many times as the trial data comes up. And one of the things that we've been recently talking about when meeting with patients and families is the fact that people have had seizures after treatment. And understandably, that's a concerning thing. And so the question is, are these seizures that are these patients who had seizures before who now have seizures? Are these patients who have rapidly progressive ALD, or what's happening? So we've asked for that data, and this is a busy slide, but I could just orient you real quickly. And that of those 67 patients, there were 9 of them have had seizures after the time of infusion of the gene therapy product. And so when it says serious, this was a question that the physicians got to decide. And so put this in there. So, take it all with a grain of salt. So what a physician and what a family think is serious may be different things. What time those seizures happened, or how many days after treatment. And you can see some of them are quite a bit out from there. Time to last seizure. So if they had the seizures, were these seizures that were continuing over time? And so even without digging really into that data, I think just to say that when we looked at this, there were patients, you can see of those 9 patients, 3 of them had recurrent seizures, whereas 6 of them had isolated seizures. So perhaps a seizure in the setting of an infection, a seizure, in the setting of a fever. And so this too is going to be data that we're going to dig into over time and hopefully present in more detail in the future. So the question, and this is a slide people have seen before, but just to try and wrap it up and put it in context, I think we all know that we wish there were better therapies. We wish patients who had cerebral ALD didn't need to get chemotherapy or have transplants or to have gene therapy, and we wish it was easier. These are hard questions. They are intense therapies in really challenging situations. And we talk about sort of making this as decisions as a balance. And so for some families and for some patients, one of those things may be more favorable. So, when we think about gene therapy, there is the MDS risk. And so that is obviously a concern. Obviously, that's a strong negative. But then there's also, are you able to find a donor? Or what is the risk of graft versus host disease? And what does that look like? And so then compared on the allo side, you don't have that MDS risk. There is a risk of cancers from chemotherapy that can occur with both of these processes, long term. But you don't have the same MDS risk that we're seeing, but you have risks of graft versus host disease. We know that there are families who really have a very difficult time finding an appropriate donor or finding an appropriate donor in real time. And so I think you have to balance those things. And so just at the end of it, I think we all are trying to do our best to take the data that we have and what we know about from allo transplant being done, for decades, comparing that with the data that we have from the clinical trial, and also what we're learning, from patients who are receiving it after FDA approval, and then the patient, parent, and caregiver preferences. And that cannot be valued enough. We have to think about that piece, because for some families, if you've had another child who received a stem cell transplant and had complications, or that went perfectly, transplant means one thing to you. Or if you know someone who had challenges with gene therapy, it means something else. So when I think about these conversations and talking to patients and families about that, it's all of those things. And it's really hard to make that decision, of course. And it's also, we can't understand because we're not in your shoes, but it's really hard to have to make that decision quickly in patients who may have had, converted to either be a later presentation without family history, or people who are their disease is moving quickly. So I think, questions have been, should this be approved? Should anyone get this? And I think the answer is that patients and families deserve options, and providers need to explain as best we can the data that we have. And with that, Paul, I'm going to turn it over to you. Stop sharing the slides so that people can see each other.
Thanks, Christy. So I'm Paul Orchard. I'm a pediatrician of a bone marrow transplant person here at Minnesota. And I appreciate the opportunity to talk with the group today as well. So I'd just like to give maybe my 10,000-foot view of what we currently know. And it's important to understand that all this data is kind of an evolution. We're getting more information all the time, essentially. But there's two big buckets that we think about these things in terms of therapy. So one is the safety one, and we've been talking about that in terms of the myelodysplasia and leukemia piece. And the other is the efficacy piece. I mean, how well does this work? And then you compile those together. And if we're going to be comparing this to transplant, then putting those things into context makes sense. So in my view, on the safety side, I don't think there's any question that auto gene therapy approach in the first 3 months, 6 months is safer than allotransplant. I don't think there's any question of that at all. Nobody's died going through the auto lenti gene therapy study. So there's been 67 kids treated. We treated 21, I think, here, but no one's died. The peri-transplant mortality, meaning the patients that die of the allotransplant procedure, is going to be in the 10-15% ballpark and potentially higher depending on what the situation is. So, like I say, I don't think there's any question that the early safety piece, that gene therapy is superior. What's becoming a little bit more harder to compile as part of this is the late safety pieces, because that's the myelodysplasia side and the leukemia side. And so, now there, as Rich and Christy presented, there's 5 patients. The N again is 67. So if we, look at that together, it's about 7% of the kids have gotten myelodysplasia now. So maybe it's going to stop there, or maybe other kids are going to get it. We just don't really know. So the late safety piece, I think, is a bit harder. The efficacy piece, again, we're still getting more information. From the data that Christy was showing, it looks like there's maybe a bit more progression in terms of the MRIs on the kids that were on gene therapy as opposed to allotransplant. But the MRIs are less important than how the kids are actually doing. There's several different clinical aspects that we look at. There's the major functional disability piece, and that's helpful statistically because that's a plus minus thing. You either have it or you don't, and that makes it easier in terms of the statistics part. But to get to major functional disability, you are very disabled. I mean, those kids have had a lot of progression of their disease. There's a neurologic functional score, and Christy showed some of that data as well. And that's whether you're symptomatic, essentially. So for instance, you get a point for seizures, but if you're having issues with understanding language, if you're having issues with being able to communicate, or those types of things, you get points for those types of things as well. So we're still looking at that data. Whether there's statistical differences between transplant, allotransplant, and gene therapy is that hasn't reached statistical significance yet. So at least as best we can tell, those things look like they're the same for now. One of the pieces we really want to look at, we don't have all that information yet, is the neuropsychology evaluations too. So if you're not symptomatic, meaning you're not having visual problems or hearing problems or those types of things, you can still be quite affected by your disease. And some of those kids have issues with processing or memory or other types of things that can be lifelong problems for them and potentially could influence whether they're going to be independent down the road or what kind of care they might need. So from a safety, just kind of put those things together, I think clearly early safety, the gene therapy is head and shoulders above allotransplant. I don't think there's any question. The late complication, most of the issues that the kids get that are life-threatening with allotransplant are early. There are kids that have problems that are long lasting in terms of chronic graft versus host disease or those types of issues, or chronic infections that might play a role later on. But most of the risks of death, for instance, with allotransplant are relatively early, whereas with gene therapy now, those things are somewhat later. So, where do we come down on these things ultimately? Again, early safety favors gene therapy, but we don't know that gene therapy is actually more effective than allotransplant. There may be some sense that it may not shut the disease down quite as fast as allotransplant. So, those things all have to get factored in. If there's been 4 kids that I'm aware of that have, for instance, progressed and have been taken off or gone on to allotransplant for progression too. So that's an issue as well. And if we put the 5 kids with MDS and the 4 kids that have progressed, so they've gone to allotransplant, and divide that by 67, it's about 13%. So that's the benefit of gene therapy and over and above allotransplant is ultimately the calculation that we need to make. And those things have gotten a bit narrower, I think, with these more recent cases. So, who does it make sense to offer gene therapy to versus allotransplant? I think it's going to be on a patient by patient basis. I think if there's a kid that's very early in the course of his disease, doesn't have good donor sources, I think absolutely that would be a patient that we consider for gene therapy. If you have a patient that's got a less score of 8 and they have a match-related donor, I think very likely most of us would consider doing an allotransplant in that sort of situation. But a lot of the situations are going to be in the middle there. And so I at least from my view, assessing what the situations are and for an individual patient and weighing the pluses and minuses of both therapies is really what we should be considering doing. So I'll stop there. And I know there's quite a few questions, and we'll do our best to answer those.
So I'll start with the questions that came in prior to tonight. So there was 5 questions. The first one is, my nephew is 4 years old and after gene therapy. So far, all his labs are fine. Is there any chance after 4 years of developing MDS, is this risk for life, or after a few years, you are no longer at risk?
Rich, do you want to do that one?
Yeah, I can do that one. I was thinking it would be good though for either you or Christy to answer that question, because I think it's an unknown right now, and I think it would be helpful for you as the treating physicians to opine on that.
I can try. So this is going to be an unsatisfying answer, but we know there is a risk after 4 years, specifically because one of the 5 patients presented later, so presented around the 7-year time point after treatment. Unfortunately, the first patient we have is now 10 years, not MDS. The first gene therapy patient is 10 years after treatment. And so we don't have decades of experience to say whether that risk changes over time or not. So it's an unknown. Of those patients, though, of the 5 boys who were diagnosed, one of them, the patient I just referenced, was significantly later than the others. The other patients had either questions, some things or hints that may have been making people think about this, or some patients did not, but all of those patients presented, 3.5 years or less from gene therapy. So there does seem to be a cluster of time. And again, this is small numbers of patients where there's a higher risk, but that is the data that we're going to try to get over time. So I wish we had a better answer than that, but that's what we know right now.
And question number 2, for children in studies 102 and 104, have you seen any disease progression? And if so, what steps can be taken to stop the progression?
Well, again, I'll ask Dr. Duncan or Dr. Orchard to answer that question. I think Dr. Duncan showed some of the data that we have, the latest data about, at least when it comes with respect to the MFDs, that we have not seen later on.
I'm happy either way.
Well, so I would just say, so the MFD again is a very late finding. The kids have to have progressed pretty much dramatically to get to the point where they got a MFD. So that's I would tend to not use that as probably the best criteria, frankly. But so the answer is yes. I mean, some of the kids have progressed, and some of the kids really have not, and I don't know why that is. But there are certainly kids that have progressed to the point where we've taken 3 off study and done an allotransplant because we were concerned about the amount of progression that they've had. But why those particular kids had progression and others did not, it's not clear to me. So, and again, we tend to see some progression with the allotransplant as well, but probably, at least the data we have now suggests that there's potentially some more progression with gene therapy, at least as a group, than we see with allotransplants.
And I think it's hard in Paul, because obviously those are the patients that you've cared for, to know sort of where that progression falls. So I think you're right. When you talk about major functional disabilities, that's quite significant progression. And that's absolutely, it's not subtle, but then whether there's progression on MRI versus progression in the NFS, there certainly have been some patients who've had clinically meaningful progression. And then there are boys who've had the progression on the MRI that hasn't turned into functional changes. So yes, but again, as we said, still data we're waiting to get with more detail.
Yeah. I mean, it makes it hard because we don't want kids to get symptomatic. Right. And so, when does it make most sense to pull the trigger to potentially say, we're worried enough about progression in this individual patient that we would offer allotransplant rather than watch them potentially progress some more? Because we've got quite good neuropsychology data that as you progress in terms of your LESS score, you have more and more functional issues, even if you aren't having vision problems or hearing problems or seizures or whatever. So we don't want to put kids through the additional risk of allotransplant, clearly, but if it looks like they're progressing over time and that course is quite clear, we would certainly consider offering allotransplant in that scenario.
And just so everybody knows the full numbers, the cases that the boys that Paul was talking about, there were a total across both studies so far of 3 failures and of boys that ended up needing an allotransplant. So it's 3 out of the 67 who ended up in that situation.
And it's for progression rather than for the MDS plus the additional.
Correct.
Yeah.
Okay, and then this question is for Rich. I would like to know where the LTF 304 data is available. About MFD and gadolinium, I always hear that at 24 months, all children are free of MFD. I would like to know the data on MFD and NFS after 24 months until today, and if there are any children after 24 months with GAD and until when. GAD enhancement, I should say.
Yeah, no, thanks. And just that's an important question. And at month 24, we know that 29 of 32 children were MFD free. And I think that's the number to keep in mind. With respect to the other measures, we continue to monitor and follow up through, including in study 304. Most recently, that was presented by Dr. Eichler, who presented those data last year. And that the one thing we're continuing to do is to reanalyze the data. We have to do another data cut, and we anticipate that we are going to publish those data within the next 6 months that show what the updated data look like.
And then for children with leukemia, if they develop a disease during the BMT, for example, diabetes or other, or need additional help, such as physical therapy or something else for a long period, who pays for the medicines for life, as in the case of diabetes, and who pays for therapy?
Yeah, this is a pretty specific question about a specific patient and their insurance, I would say. And this is something where we'd encourage you to discuss this with your treating physician. And if a conversation needs to take place, we could always be reached at My Bluebird support, especially for patients who get treated with Skysona. Patients in the clinical trial, it might be a little different. And, that's one where you speak with the investigator, the physician who's taking care of you, and they're in contact with us all the time too.
And then if a child has leukemia after BMT, the leukemia goes into remission or the child is cured, is there a possibility that this disease will return again in the future?
Can I do that one first?
Sure. Yeah, I'm happy to answer that question. So the short answer is yes, there's a possibility. The data that we use for MDS cases or the myelodysplasia cases, which is what those boys are getting the transplant for, it is based on children who have MDS for other reasons. So people who've had leukemia, who've gotten cancer treatments or other things before. And so it's a little tricky to do that comparison because obviously those children are different than boys with ALD who didn't get chemotherapy prior to their treatment. So there is a possibility. If we try to, take that data and make it fit ours, is it possible? Yes, the survival data, if you try to do it as best you can for children with myelodysplasia who get allotransplants, is quite good. But we do monitor patients with bone marrow tests and things, for a lifetime, basically, or for at least, many years after that treatment for MDS, whether it's related to gene therapy or children who get myelodysplasia or MDS for some other reason. We can tell you that that's not been the case with the first 3 boys, but there's not enough. It's not enough time to tell you how meaningful that will be, because those cases presented in 2021.
Okay, and now we'll turn it over to the questions in the chat. From Kirsten, my understanding is that the enrollment for ALD 304 was by invitation only. What was the criteria for choosing who would be involved?
So every patient who participated in ALT 102 or 104 was offered participation in 304. So the hope was that everyone would participate. And so we would be able to get data, going long-term, most importantly for the safety and the health of the patients, but also so we could learn more. So if you got treated on 102, 104, you were eligible for the long-term follow-up protocol.
And then another question.
I just want to add, similarly, for anybody who's treated with Skysona, sorry, Elisa, for talking over you, but we have the registry program where everybody's invited to be part of that who's been treated as well. So we're in that study, patients will be followed up for 15 years as well. So it's, again, thinking about the safety and what we're going to learn as we can move on into the future.
Right. And that's something that the FDA in the United States and similarly in Europe mandates. Anyone who gets gene therapy, not just for ALD, but for any disease, the expectation is that you are followed for 15 years. And so that's not something anyone, if we decide not to do a long-term follow-up protocol, any of that, it's not an option. You have no choice. And we all support that people should be followed that long, if not longer.
How many cases of neurologic progression have occurred beyond that would typically happen post-transplant while engraftment of the brain occurs? What is the threshold for reporting this progression?
Well, maybe I'll start there. So we keep track of the LESS scores, which is the MRI-based scores. So that's one means of looking at progression. And then the kids have neuropsychology testing done. We just don't have that data compiled to be able to share with people. And then there's the neurologic functional scores and the MFD things that we were talking about before. So all that data is collected, all that data is available. So it's still evolving. So how different it's going to be than allotransplant is something we're still looking at. But there's no defined characteristics outside of the ones we just kind of talked about in terms of progression. Say if you got to a 3 or you got to a 4 or whatever in terms of a LESS score that yeah, sorry, a less score, that we would say that that kid has progressed. I mean, they're all those numbers just sort of evolve.
And this is just a comment from Katie Becker. I'm not sure if this was on the slide, but we have seen seizures post both types of treatment. Given there is an old scar, boys do have a higher risk for seizures, although for the vast majority of boys, that risk is very low. So I guess what she's saying is that there is risk for seizure for both BMT and gene therapy. Any comments on that?
Yeah, I think without question that's true. That the bigger picture question is, are they different? And we haven't really looked at our data to explore that very well. I don't know, Christy, if you guys have, but that's not been a endpoint in terms of our analyses and on the allo side, but certainly something we can look at.
Yeah, I think it's a good idea to look at it, in more detail. We haven't looked at it specifically, but have certainly seen it in both cases, or both types of treatment, I should say.
Thank you. And then this is from Rochelle. First, I would like to say that we are still grateful for Bluebird Bio gene therapy and our providers. Secondly, my question is, I understand that when it comes to cancer, the donors can be less of a match, considering that GVHD can help fight the cancer. Are there any other reasons for this? I'm not sure if I understand that question.
I think perhaps the question is sometimes if you have a cancer, and so I think there are two things. So one, if you're trying to find a donor for someone with ALD, you certainly want to try and make sure that the person who's the donor does not have that. And so, that risk always exists. So sometimes it's hard to find a donor. It's true that sometimes there's something, the graft versus host disease is the case where the person who's getting the treatment, their cells attack the person who's having the transplant. And that can be life-threatening, life-altering, or minor. And there's a huge range. That same thing, which is a negative reaction when you get it, if you have cancer, it can also fight the cancer cells. So sometimes we do tolerate a less well-matched, someone who's less well-matched in that case, because you might be getting that benefit. We would never do that intentionally. You always want to find the best match you absolutely can. But if you get in that spot, with MDS, or you get in that spot with leukemia, we look at it through a slightly different lens.
Next question, what prompted the change in conditioning regimen between ALD 102 and 104? Was there any correlation to the cases of MDS thus far? As a commercial product, regimens will vary. Will this possibly have any impact on possible MDS cases going forward?
I'll start and then hand it over to Dr. Duncan, because I know we've talked about this as well. The reason there was a change made between those two studies was, at the time, fludarabine was being used as a conditioning agent for transplant because it had fewer side effects than cyclophosphamide. And so the idea was it made sense that we could use that in order to determine maybe there would be less side effects when we went with a conditioning regimen of bu-flu compared to bu-cy. And so that's where the 104 change occurred. And then Dr. Duncan.
Yeah, that's true. It's and I think the important thing, and one of the things that may be coming up or part of this question, is about the MDS cases. And just to say that, of those two, there were the two studies. The first study, patients got the busulfan, the cyclophosphamide chemotherapy, which is more intense, or a higher degree of intensity of chemotherapy. And that's important for short-term toxicities, but also for long-term toxicities, or the long-term complications you can get after allotransplant. So the move was intended to be one of better safety. And so less toxicity, better safety. That first regimen of busulfan, cyclophosphamide, those two chemotherapies, was what was done in the first boys who got this treatment, or similar treatment, not the same product, in France. So that's where the original came from. And so the movement to fludarabine was to try, for boys who don't have cancer, to try and use a less intense therapy. What we're seeing now, and this is obviously still an evolution, is that the boys treated on the first case, and again, at the first study, those patients who've now been followed longer, the patients who are 10 years, almost 10 years out from treatment, there's been one case of myelodysplasia. And then in the patients who got the lower intensity chemotherapy, there are 4 cases. So it seems, and obviously this is data that we still need to try to understand, and there are some reasons we think perhaps that could happen, that what we thought at the time and with the community, and there were representatives from all the transplant, centers, the big ALD centers having these discussions, was that this protocol seems to have a higher risk, although those numbers would never meet any sort of statistical thing if you actually got the less intense chemotherapy. And so you're right that the FDA label does not say you have to give a certain type of chemotherapy, and so that the centers are free to choose. I do know that the centers, however, being representing one of them, have talked about this and have had conversations, and I know that so my center, just to be completely honest, we've done busulfan cyclophosphamide because of that longer duration of follow-up and because of this potential observation. So for the boys who've gotten the commercial therapy. So I think what is going to be important is that the centers, we all continue to talk to each other so that the data that comes afterwards is meaningful, can be compared, and that we're all trying to offer our best practices for safety. And, it's a small number of centers. We all know each other. We all get along. And so my hope is that transparency between centers and that collaboration will continue. So what's described, different risks of MDS and different things, doesn't actually happen.
Right. So I think it's important. Yeah, I think that observation is clear that there was one case in 102, there were 4 cases in 104 at this point. However, we don't really have an understanding yet of what those differences are. And one of the things that we're going to be doing is we're pulling in the investigators and other experts to have this discussion and try to figure out where we go from here with that information, what it could be, how it could be meaningful. But at this point, we really don't know if that is the difference, and we'll keep working on it to find that out.
Yeah, I would just add that we've been using busulfan and fludarabine for 10 years or more on the allogeneic side and haven't seen any issues. So, I mean, obviously that's an allo transplant versus an auto transplant, but it's also important to understand that the cells that are becoming myelodysplasia or leukemia did not see the chemotherapy, right? So those cells were removed, those cells are manufactured, those cells are frozen down while the kids are getting the chemotherapy, and then the cells are being administered after. So, would the immune function of the cells after getting busulfan and cyclophosphamide be different than busulfan and fludarabine? I suppose hypothetically, but I don't know. And I think so, and Rich, you can correct me on this, but I think, wasn't there 17 kids? No, how many were on 102? 20 some?
No, it was 32 in 102 and 35.
Okay, so roughly the numbers are about the same, right? I somehow envisioned that maybe there's fewer kids on 102, but not.
I think we started at 17 and then we started at 17 and kept going.
Yeah, right. So 17 in the first paper, I think maybe that's what the story was. So, but so it's hard to know if the chemotherapy piece makes a difference, but again, we've had a lot of experience with busulfan and cyclophosphamide, or busulfan and fludarabine, and haven't seen anything on the allogeneic side.
And just a comment from my past experience, which was 11 years ago, the hardest decision that we had to make as a family, and this is something I brought up at the March meeting, was this exact question. One of the institutions we were going to was using low intensity chemo. The other one was using high intensity chemo. And when I asked the questions, they both made sense. So it was a really hard decision to make for us, especially when we were under the gun, to have to go through therapy right away. So just, a note to the doctors and everyone on this, if we can just really work towards a consensus on the chemo regimen, I think would be amazing for the community going forward. Yeah.
Well, I don't think that's going to happen, but so we're going to use the same conditioning regimen for the gene therapy patients as for the allo patients, so we can compare them head to head. So you've got over 10 years of experience using this preparative regimen. We know what it kind of does. And I think if we really want to sort out whether allogeneic or autologous approaches seem to be better, being able to compare them, I think, is going to be helpful. So that's going to be our approach, at least.
I understand. Okay, next question. It has been expressed that gene therapy takes a little longer to halt progression than allo transplant. Is there any data as to how much longer this takes?
Yeah, I don't think we have data that supports it does or does not at this point. Correct me if I'm wrong, Rich, or Paul. I think people have said that. Some people have said it's faster, some people slower, but I don't think we have data that actually says that.
I missed that question. Faster or slower than?
So if you compare the stop of the progression with gene therapy compared to the stopping of the progression with allo transplant, is one faster than the other? You know, do we know? And if so, what's that data and why?
We're going to be looking at some of those things. We're looking at biomarkers and other things that might give us some sense of this as well. And we didn't really talk about, so we've been talking about the less scores in terms of how the kids have kind of done, but we haven't really talked about the gadolinium piece. So the gadolinium positivity suggests that you got active cerebral inflammation. And I think it's pretty clear that it seems to flicker more on and off in the gene therapy patients than in the allo side of things. But that's kind of a binary outcome. It's either there or it's not. It's not very quantitative, which is a bit of a problem, because you may lose 90% of your gadolinium enhancement, but you might still be called positive. So that data is kind of hard to sort out. But
And next question. Well, there was one patient on 102. What theories are there to why the cluster on 104? Is the conditioning, okay, another question about conditioning. Is the conditioning being looked at?
The conditioning is being looked at. There are other things that we may have that were done slightly differently as far as more patients got a certain drug for mobilization. There's some small other changes, but the biggest difference was the conditioning. So we're looking at that. We're looking at, or as best we can with the data, and how the people's immune systems recovered. We don't have that in like real great detail, but we're also thinking about whether the centers have collected additional data. So do patients recover faster with one than the other as far as their immune system? Trying to think about anything that was different. And we haven't, the biggest thing, obviously, the most obvious change is that chemo, but as Paul said, we don't know that that's what it was. It's just the biggest difference. So we're trying to go over those things in as much detail as we possibly can.
yeah, and the numbers, as Christy mentioned before, are so small that it's pretty hard to tell. It's not clear there's a difference, I guess, at this point, but we may know more later.
Got it. And we are at time. So is there any final comments from Dr. Duncan, Dr. Orchard, Dr. Colvin, anyone else?
I think I just wanted to thank the community again for coming together tonight, for talking about those things. Encourage you to reach out to any of us on the call, or other people, or if you're from different parts of the country and want to be connected with people, please reach out.
Yeah,
Yeah,
I
I
would
would
just second that. Whoops, go ahead, Rich.
No, go ahead, Paul. You beat me to it.
I was just going to second that. If there's, I'm sure there may be more questions, or people are going to think about it later and have questions later. So please feel free to, ask those. We're not hard to find. So.
Yeah, and thanks, Elisa, and thanks for all of you for being here today, for sharing this time with us. And we want to just reiterate that, one, our commitment to the community, but also especially we're going to keep publishing, we're going to keep sharing information. And again, thank you for being here tonight.
yes, and I just wanted to say thank you all. Thank you to the doctors. Thank you, Bluebird, for being part of this, and everyone in the community. And I wish it could be something as simple as take Tylenol and your fever will go away. But as we know, ALD is not that simple, and what works for one boy might not work for another. And we're just grateful to have more than one therapy available for our boys, and that's still a huge gift. And, again, just make sure to talk to your doctors if you do have any questions. And if there's anything that you still need to know, reach out. We will try to get you the answers when we can. And yeah, it just takes all of us as a community to work together and to have this information shared. So thank you all.